Namaskar. Today we will study deoxyribonucleic acid or DNA. So it is a type of nucleic acid, and we can have two types of nucleic acid, deoxyribonucleic acid and ribonucleic acid, based on the sugar. And this nucleic acid is a polymer of nucleotides. We can say it is a biopolymer of nucleotides, and nucleotides consist of a sugar, one base and one phosphate. So the sugar is deoxyribose in case of DNA, and the nitrogenous base can be either adenine or guanine, cytosine, thymidine, and the nitrogenous bases can be either purine bases or pyrimidine bases. So adenine, guanine, the purine base, cytosine and thymine, the pyrimidine bases in DNA. Now, out of these two purine base, adenine and guanine, and two pyrimidine bases, thymine and cytosine, one base will be there attached to this deoxyribose sugar, and to this deoxyribose sugar the phosphate will be attached. So it is a chain of sugar phosphate. So at one end there is this 5' position, and the other end it has the 3' position of the sugar. So let us see the sugar, this deoxyribose sugar. This is the structure of the deoxyribose sugar: 1', 2', 3', 4', and this is the fifth carbon. So what happens at one end to this hydroxy, the phosphate is attached, and the other end to this hydroxy, the 3', one phosphate is attached. So 5' to 3' direction the chain will be elongated, and the to this 1' position the bases are attached. So you can see here one nucleotide here, deoxypentose sugar, the adenine base here. So this is the nucleoside deoxyadenosine with a phosphate, and so a chain of sugar phosphate is there. To each sugar one base is attached, and as we know the DNA is a double helical structure. One strand is 5' to 3' and the other strand is 3' to 5'. So 5' to 3' one strand, 3' to 5' one strand, and though it is the helical structure, we have shown here how these are linked. Suppose here in one strand adenine is there, and in the other strand thymine will be forming the bond. So here we have shown it: AT, TA, CG, GC, CG, like this. And how it is synthesized? From one DNA we can have two DNAs. So first there will be unwinding of the two strands by the enzyme DNA gyrase. So there will be a separation of the two strands, and each of the strand will become a template for two DNA synthesis. So from one DNA we can have two DNA synthesis, and the enzyme here is DNA polymerase. So this strand, the complementary strand will be synthesized. For this strand, another complementary strand will be synthesized, and we can have two DNA from one DNA. But during the RNA synthesis, this 3' to 5' strand, which is called the antisense strand, will be acting as a template, so that the sense strand or 5' to 3' strand of the DNA will have the same gene sequence as that of the messenger RNA. But the thing is, instead of the thymine, there will be uracil in messenger RNA. So you can see here, suppose this is the DNA, and here the sugar phosphate like this is there. So these are the bases. So we have shown here the bases, and you can see the 5' to 3' strand, this is called the coding strand or the sense strand, have the same sequence as that of the messenger RNA. ATG, so AUG, instead of T there is U. Then ATC, instead of T there is U. Then TCG, so instead of T there is U. So the messenger RNA will have the same base sequence as that of the coding strand or the sense strand. Only difference is, instead of T in the sense strand, we have U in the messenger RNA. And how this happens, the other strand, which is the anti-coding strand or template strand or anti-sense strand, that acts as a template for the synthesis of messenger RNA. So we have this antisense strand, and the complementary bases are there for messenger RNA by the enzyme DNA dependent RNA polymerase. T for A for U, C for G, T for A. So to summarize, deoxyribonucleic acid has two strands, one strand from 5' to 3' direction and the other strand from 3' to 5' direction. And this 5' to 3' direction is called, the strand is called coding strand or sense strand or plus strand. From 3' to 5' direction, the strand is called antisense strand or template strand or non-coding strand or minor strand. And by the action of the DNA gyrase, these two strands are separated, and then these two strands, say this is separated by the action of DNA gyrase, one strand sense strand, the other is antisense strand. By the action of DNA polymerase, both these strands can copy the parent DNA. So from one DNA we can have two copies of the DNA, and this antisense strand can act as a template for RNA synthesis. Messenger RNA can be synthesized from this antisense strand, so that the base sequence of messenger RNA will have the same base sequence as that of the sense strand. Only the difference is thymine is replaced by uracil. So here in this DNA, the bonds, the base pairing occurs between adenine and thymine, double bond, cytosine and guanine triple bond. So this is all about DNA. Thank you.